I had the privilege of presenting the first data disclosure of the phase 1 trial of the first-in-class combination of the WEE1 inhibitor, Zedoresertib, plus the PKMYT1 inhibitor, Lunresertib, in patients with advanced solid tumors harboring CCNE1, FBXW7, and PPP2R1A genomic alterations. Cancers with these alterations represent key areas of unmet clinical need and include tumor types such as ovarian cancer, uterine cancers, bladder cancers, and lung squamous cancers. We know that CCNE1 amplification induces high replication stress through premature S-phase entry, and FBXW7, which is an E3 ubiquitin ligase, when we see inactivating FBXW7 mutations, these increase cyclin E1 levels and replication stress. In addition to that, PPP2RNA hotspot inactivating mutations increase replication stress. And therefore, cancers with these different alterations represent areas of unmet clinical need with no available standard of care options currently. We also know that cyclin E1 regulates the G1S phase transition, whilst FBXW7 proteolytically degrades cyclin E1, and therefore, as mentioned, when FBXW7 is mutated, cyclin E1 is overexpressed. And the whole rationale for the study is that WEE1 regulates S-phase replication origin firing and G2M checkpoint phosphorylation of CDK1 at tyrosine 15, whilst PKMYT1 regulates the G2M cell cycle checkpoint progression through threonine 14 phosphorylation of CDK1, therefore causing cell cycle arrest. And cells under high replication stress are more dependent on the G2M transition for DNA damage repair. Both Zedoresertib and Lunresertib are currently already in phase 1 clinical trials and have demonstrated manageable safety profiles at the recommended phase 2 doses as monotherapies. Zedoresertib is a potent, selective, and brain-penetrant WEE1 kinase inhibitor, whilst Lunresertib is a potent, selective PKMYT1 kinase inhibitor. And we combine these because WEE1 was identified as a top synthetic lethal hit in PKMYT1 inhibition chemogenomic screens, and the combination was highly synergistic at sublethal doses. And the rationale behind this, we believe, is that WEE1 inhibition exacerbates CCNE1 amplified induced replication stress during S phase, and at the G2M checkpoint, WEE1 and PKMYT1 inhibition reduces CDK1 phosphorylation, causing hyperactivation, and cells carrying unrepaired DNA damage are then driven prematurely into mitosis, leading to mitotic catastrophe and cell death. And importantly, we have observed strong synthetic lethality in vivo with compelling synergy leading to durable regressions in multiple tumor types, including those with CCNE1 amplification, cyclin E1 overexpression, FBXW7 mutations, and other key alterations in a range of different tumor types. And that provided a rationale to proceed to this combination. We used a Bayesian design to dose escalate through multiple different dose levels of the combination of Zedoresertib given daily, together with Lunresertib given on a 3 days on, 4 days off schedule. The eligibility were pretty standard for a phase 1 trial, but we mandated that all patients had to have either CCNE1 amplification or FBXW7 mutations or PPP2R1A mutations. The primary objectives included safety and tolerability of the combination, establishment of the MTD. Secondary objectives included preliminary antitumor activity, pharmacokinetics, and pharmacodynamics of the combination. In total, we treated 62 patients, median age of 61 years. There were more females than males because of the types of cancers we treated. All patients had an ECOG performance status of 0 to 1. This was a heavily pretreated, resistant, and refractory cancer population with a median of 3 prior lines of therapy and up to 9 prior lines of systemic anti-cancer therapies. 73% of patients had at least 3 or more prior lines of therapy, and 89% had prior platinum chemotherapy agents. The most common tumor types included ovarian cancer, 42% of these patients, and 92% of these ovarian cancer patients had platinum-resistant or refractory disease. We also treated colorectal patients and pancreatic cancer patients, and all of these patients had one of the 3 alterations. Overall, we found that the combination was manageable and in line with either drug given as monotherapy. The most commonly reported treatment-related adverse events included grade 1 to grade 2 GI side effects, rash. No patients, importantly, were discontinued in terms of the drug treatment due to any treatment-related adverse events. In terms of the PK exposures, Lunresertib exposures and Zedoresertib exposures in combination were in line with monotherapy, and to date, no signals of drug-drug interactions were observed, leading to exposures that were different with monotherapy. We observed robust target engagement of Zedoresertib and Lunresertib in combination across multiple dose levels. We saw phospho-CDK1 reductions in paired tumor biopsies. And we also observed DNA damage induction observed in a majority of patients with increases in gamma H2AX. Importantly, in terms of efficacy, we observed frequent and deep tumor shrinkage with the combination in heavily pretreated patients across multiple different tumor types. Overall, 51% out of the 51 patients with target lesions showed tumor shrinkage, and 10 patients achieved a response. For example, 7 patients with CCNE1 amplified ovarian cancer, 1 patient with FBXW7 mutation ovarian cancer, a patient with HR-positive, HER2-positive breast cancer with both CCNE1 amplification and an FBXW7 mutation, and also a patient with metastatic colorectal cancer with an FBXW7 mutation responded. And the overall disease control rate was 68.5% in all patients. And approximately a third of patients remained on treatment for at least 16 weeks. When we then look at the resistant refractory ovarian cancer population, we treated a total of 24 patients, and the overall response was 37.5%, and the disease control rate was 87.5%, with 80% of these patients experiencing a tumor shrinkage. In addition to that, when we then look at the CCNE1 amplified resistant refractory ovarian cancer population, we observe that the overall response rate is further enriched and is 42.1%, and the disease control rate is 94.7%, with approximately 81% of patients experiencing tumor shrinkage. And these efficacy data are supported by the ctDNA molecular response rate of 44% observed in all patients across all dose levels. To conclude, we have shown the first early clinical proof of concept for the synthetic lethal combination of the WEE1 inhibitor Zedoresertib plus the PKMYT1 inhibitor Lunresertib in multiple different tumor types with CCNE1 amplification, FBXW7 mutations, or PPP2R1A mutation cancers. The combination was well tolerated and demonstrated strong anti-tumor activity, particularly in patients with resistant or refractory ovarian cancer. Responses were observed in patients following progression on or after ADC treatments as well. Currently, schedule and dose optimization in multiple different tumor types is currently ongoing, and therefore this oral combination may provide a novel therapeutic option for patients harboring these key alterations across multiple different tumor types with great unmet clinical need. Based on these data, this combination of Zedoresertib plus Lunresertib has been granted FDA Fast Track designation in patients with ovarian cancer harboring CCNE1 amplification, FBXW7 mutations, or PPP2R1A deleterious mutations. Thank you very much.